Welcome to this live stream. I'm delighted this morning to be with Professor James Shapiro again. How are you doing, Professor?
I'm doing fine. How are you?
I'm very well. So you've written a new piece, which is really interesting. It's on chromoanagenesis, and we're going to be speaking about this today. So to begin off with, the easy question, what does chromoanagenesis mean?
Well, chromoanagenesis is a Latinate word that means chromosome rebirth. It was coined to describe massive chromosome rearrangements, restructuring events that occur at multiple sites in the genome within one or two cell generations. The idea of rebirth is that multi-site chromosome restructuring often creates a genome with a new and different way of expressing the information it contains. This kind of rapid genome transformation was discovered 13 years ago in cancer cells, which are known to be highly unstable. Nonetheless, within a year, the same kinds of rapid genome restructurings were shown to occur in the genomes of human beings who display the characteristics of people who carry genetic abnormalities. Chromoanagenesis was also found to occur in plants and animals, as well as fungi. So this phenomenon in higher organisms for approximately 2 billion years. There are at least 3 different patterns of chromoanagenesis, all of which are seen in multiple organisms. That tells us that the dispersed changes do not simply result from random breaks in many chromosomes, which are simply repaired. Instead, the 3 distinct patterns mean that each type of chromoanagenesis reflects the operation of particular systems that cells have for inducing and repairing chromosome damage.
Brilliant. There's a follow-up question to that. Why are massive chromosome rearrangements important for evolution?
Well, massive chromosome rearrangements change all kinds of things in the genome about how it's regulated and how it behaves. And so the genome is expressed differently, and that can lead to the operation of different genomic networks to make different character traits, which may affect the survival of the species. Also, changes in genome structure can lead to chimeric regions where new proteins and new products are formed. And third, chromosome rearrangements proceeding through meiosis and the production of gametes. So it leads to reproductive isolation of the new cell type or the new organism with the new genome from the parent. And that's characteristic of speciation.
Yeah. And that obviously has significant implications for the way that we view the standard way of understanding evolution. What did you discover about these rearrangements that changes our thinking about evolutionary theory?
Well, what I discovered about them is one, that they're very common. So a major evolutionary change, what we call macroevolutionary change, altering multiple traits and reproductive boundaries, can happen quite rapidly. It's not necessary to accumulate many independent mutations over long periods of time. We can observe this kind of punctuated evolutionary pattern in cancer, which is the disease of somatic evolution. It's not an accident that chromoanagenesis was first described in cancer cell genomes. Tumors can be stable for many years and then change abruptly when they evolve into a more advanced form of cancer. And that's a model for organic evolution.
Yep. Brilliant. Thank you for that answer, Professor. And so, just to key off what you said there, something which is quite interesting, you've written that structural variations in eukaryotic chromosomes occur far more frequently than single nucleotide variations. What is the significance of this finding?
Well, it tells us that there are two differences. One is that these changes occur more often, so they're more likely to be the idea that single nucleotide changes were the basis of genetic change implied that evolution was a very slow and long-term process. But the fact that we find chromosome structural variations more frequently tell us that they are more likely to be an important source of new characteristics and new properties than single nucleotide variations.
Yeah. And single nucleotide variation is obviously the standard way that biological novelty is supposed to have the fodder for evolutionary change. So obviously this is a huge, paradigm shift from that perspective. You've also written that
it shows that we're dealing with something which was not conceivable in the 20th century, even 20 years ago. People just weren't thinking that way. And now we know that these kinds of changes happen frequently. And so we shouldn't be surprised at finding them to play a role in evolution. There was even a paper put online. It hasn't been accepted for publication yet. That talks about a whole new form of annelid worms arising as a result of chromoanagenesis.
And that is completely unexpected from the standard perspective.
Oh, absolutely. Absolutely. That's the whole basis of neo-Darwinian evolution is that things occur slowly, and you have a collection of mutations which accumulate because they lead to better growth. But what this is telling us is that major changes occur quickly, and they can affect many parts of the genome at the same time.
That's right. You've also written that sometimes the terms chromosome or chromosome catastrophe are used to describe the multiple sudden genome arrangements involved in chromoanagenesis events. Do you think the usage of such terms is incorrect or misleading?
Well, I think it reflects a kind of negative point of view that somehow the existing structure of the genome is the best, and that if you change it, you're is that the changes in the structure of the genome lead to new possibilities. And chromoanagenesis means chromosome rebirth, and that's a more positive way of thinking about it. You're creating a new system for the heredity of the organism, and that can be positive and provide adaptations which the old system could not provide.
Yeah, absolutely. Now, in terms of chromoanagenesis, there's 3 classes that have been regularly recognized. What are these classes?
Well, the first one is called chromothripsis, which means chromosome shattering. And this occurs when a fragment of a chromosome is left behind at cell division and gets encased in a structure called a micronucleus, which sits on the outside of the cell nucleus. At mitosis in cells like that with micronuclei, the DNA that's in the micronucleus becomes fragmented into many different places. And then in the next cell division, after these fragments have been absorbed back into the nucleus, they get put back together again. So you get a completely rearranged chromosome. And there are many examples of this. This is the best understood case of chromoanagensis. Another form is called chromoplexy, which means chromosome weaving. And this is where you have a group of chromosomes, say 3 or 4 or 5, which have multiple exchanges between them. And they form a pattern of those 4 or 5 or 3 chromosomes. And that's a different form of rearrangement. And that has been documented a number of times as well. And then the last one is something called chromoanagensis, which means chromosome building up. And that leads to increased copy numbers of different regions of the chromosome, which become duplicated. And that changes their expression and also provides more opportunities for evolutionary change in the genome. There may be more, there may be more kinds of rearrangements. Analysis is not exhaustive yet, but those are the 3 that are recognized. And they occur repeatedly, which tells us that the changes are not just random breaks in chromosomes and rejoining. There's a cell biology and biochemistry behind it.
Brilliant. Thank you for that. Now, this is an interesting question. I had to think about this and I thought, do you know what? This is going to help us understand the situation better. Is there a role for higher level biological control in chromoanagenesis events? And what's the significance of this?
Well, the example of chromothripsis, where the genome is responding or the chromosome is responding to its presence during cell division and being encased in a micronucleus, is one kind of what happens. Another case is very interesting. Was there's a kind of chromoanagenesis, which occurs kind of chromoanasynthesis, which is called multiple de novo copy number variations and multiple increases in copies of different genetic regions. And this was discovered a couple of years ago, but it was only found to occur in a short period around fertilization of the embryo. It's either occurs during formation of sperm or shortly after the sperm enters the egg cell. It does not occur later after embryonic development begins. And so that means that it's strictly limited in time when it occurs, and it has to be subject to some kind of control.
Yeah. And it's interesting because this goes back to essentially the way that we look at biological systems. So if we look at it from a reductive point of view, that would be unexpected. But from a process ontology, or the idea, as Barbara McClintock put it, that the genome is a highly sensitive organ of the cell, then, you know, this type of process starts making sense. Can you hear me?
I couldn't, no. I just heard you talking about Barbara McClintock and the genome as sensitive organ of the cell. And then you went
Yes.
silent.
Okay. Yeah. So I'll repeat it again. What I meant is that, having this level of control at a higher biological level would make sense from the perspective of process ontology, this non-reductive way of looking at biological systems where there's multiple levels of causality, right? From a reductive point of view, a lot of these things that you're speaking about, from the reductive neo-Darwinian perspective, are quite unexpected.
That's right. And it fits with the idea that the ability to change the genome is a useful thing to have. It's built into organisms. They can change their genomes and make new things, and that's part of what we see with chromoanagenesis. I realize I left out a little bit of my answer to how chromoanagenesis changes our thinking about evolutionary change. Back to that.
Yeah.
I'm just having to get something off of my screen here.
That's
as I was recently writing a chapter on the evolutionary potential of chromoanagenesis, I discovered that most cases of double-strand chromosome break repair in the phenomenon utilize a remarkable system for joining broken chromosome ends. The usual view of repair is that two double-stranded broken ends are ligated together with modest deletions of some base pairs to make them suitable for what is called conventional non-homologous end joining. And eukaryotic cells do have a repair system that carries out this form of repair. But they also have another system called alternative end joining, which inserts new extra sequences between the broken ends when it connects them. And this struck me as a remarkable and potential source of elements in the evolving reborn genome.
right. And that's quite interesting, because that fits into the larger picture of how these processes could bring around rapid evolutionary change. You also write that the first evolutionary lesson learned from widespread documentation of chromoanagenesis is the major physical reorganization of eukaryotic genomes can take place very quickly. Please explain the significance of this.
Well, if we're thinking about genome change as accumulation of accidents, as in the conventional theory, that takes a long time to happen. If we're thinking about genome change as something that is a cellular function and can happen rapidly at different places, that gives you a whole different way of thinking about evolutionary change. And we're certain of that because we've seen it many times now. There are dozens, probably hundreds of scientific papers documenting that. And that's just a whole different way to think about modifying the genome.
Yeah. A related question, and I didn't put this in the questions, but a related question which I thought, Professor, you could explain to the viewers is, there's this famous understanding, this myth that people have because of the writings of people like Richard Dawkins, that the genome is sealed off from basically what's happening outside of the cell and what's happening outside of the organism. But clearly, from what we can see from all of these processes taking place, the genome is, like Barbara McClintock said, a highly sensitive organ of the cell.
Right. This was the idea put forward first by August, that there's a split between the germline and the soma. that never made sense for organisms like plants, where the germline, the flowers, come from somatic cells. So it couldn't apply to those organisms. The ability of cells to change their genome in a distributed way all across the genome is something that wasn't considered possible, but it happens. And as we look at it, when we see this kind of process, when we see massive chromosome changes, we find that there are other ways that it is biased towards creating more genetic novelty. I mentioned that there's a difference between conventional non-homologous end joining, broken chromosomes, and what is called alternative end joining, which is what happens frequently in chromoanagenesis. And alternative end joining joins broken ends with single-stranded extensions and requires a DNA polymerase. And it's the most remarkable enzyme, which is encoded by the Pol Q locus, and it's named Pol theta, which extends and connects the single-stranded extensions of two broken ends. And in the process of making such connections, Pol theta frequently inserts new sequences into the junction. Sometimes these sequences are short and copied from the ends of the broken chromosomes. But sometimes Pol theta carries out what is called template switching and copies one or several sequences from other sites in the genome. This is quite an amazing process into the new junction. And sometimes Pol theta polymerizes and inserts untemplated existing DNA. So the details of the way the DNA is joined together is such that it maximizes the generation of genomic novelty. And that maybe where many of the new sequences which come about through in evolution come from. And Pol theta and alternative end joining is present in all eukaryotes, except for yeast, which is a different polymerase for this. And it's, as I said, it's quite a remarkable enzyme. And it brings about this kind of chromosome joining with new chromosome sequences or new combinations of sequences in between the pieces of the parental chromosomes that are hooked together. So it's a very effective way of diversifying the sequence, of creating novelty in the DNA sequences. And that fits with a function of creating genomic diversity and doesn't fit with just a simple repair.
Absolutely. And a follow-up question to that is, this widespread documentation of alternative end joining and something that we simply cannot deny anymore because we've seen so much research on this topic. What does this mean for the wider evolutionary picture?
Well, the role of alternative end joining and sequence diversification means that eukaryotic cells can make endless combinations of existing chromosome sites and also generate completely novel sequences in breakpoint junctions. This kind of well-documented genomic innovation would have been dismissed as impossible as recently as 20 years ago. And it's a whole new process for living organisms to change their. I like to say about this is that it shows that organisms are uh, built to evolve because there will always be crises in the life of organisms, and they will always need to produce new properties. And they've continued doing it throughout the history of life on Earth. And so instead of thinking of evolution and genetic change as accidents or problems, we need to think about them as positive and purposeful events. And the living organisms are built to evolve and change. And the change is not something abnormal or unusual. That goes back also to the difference between chromosome crisis and chromosome rebirth in what you call this process.
Absolutely, absolutely. And I really love the way that you highlight the way that we use terms to describe the processes that we see. It gives away the tacit assumptions that we have about what we're expecting to see. So if you call it a crisis or catastrophe, it means that you think evolution is going to work in a very slow way, in an unintelligent way. But clearly, if there's rearrangement going on, there's chromoanagenesis going on, there is alternative end joining processes in place, then this is not a catastrophe. It's simply a chromosome rebirth, right? And we should
Right.
be exploring it from that positive angle.
Exactly. Yeah. I think once we realize that evolution is built in, genome evolution is clearly built into the genomes of living organisms. They have all these ways of changing their DNA. And we see them used time and time again and creating novelty. And of course, not all of the novelties work, but the novelties are such that they have a much better chance of being successful than just accumulating individual mutations. And the question that's open now, is there any kind of control over this process? And people would formerly have discarded or turned down that idea as being vitalistic. But I think now that we know about how to induce cells to make these major changes, we can begin to investigate that. And we can ask, do we see certain patterns? Do we see effects of conditions on these changes? And we begin to define rules which indicate to us that this is not just a random process, but that it has some direction to it. And we have to provide good empirical evidence for that to convince people. But I think that that's in the cards.
Absolutely. And if I was to summarize our discussion, could we say there's three main takeaways? The first takeaway being that the source of variation is not just accidental mutations. You can have, this chromoanagenesis processes going on, and also the speed at which this is happening is far faster than we originally thought, and that all of this is not just happening without higher level causality within the organism.
Yes, the same thing is true of transposable elements. Their behavior is sensitive to the conditions of the organism, and they can become activated by ecological events or experience. And they play a major role too in changing the genome. And they're not limited to action at a specific site in the genome either. So between chromoanagenesis and mobile genetic elements, I think we see that there's a whole vast toolbox of ways that organisms can change their genetic endowments rapidly, and those processes can be subject to feedback from the life history of the organism. And I think that's a major change in the way we think about evolution.
Absolutely. One of the things that you wrote in your famous book, Evolution from a View from the 21st Century, this is the first edition, is that given the status of biological evolution, we can anticipate that a paradigm shift in our understanding of that subject will have repercussions far outside the life sciences. How such an evolutionary paradigm shift will play out in the physical and social sciences remains to be seen, but it is possible to predict that the cognitive, psychological, and social sciences will have an increased influence on biology, especially when it comes to the acquisition and processing of information. So, to end this, interview from a big picture perspective, again, this is just something else for maybe the third edition of A View from the 21st Century in terms of how our entire picture is just going through an entire shift.
Well, there's a big debate to how intelligent and how living organisms and living cells are. I wrote a paper in 2020 called "All Living Cells Are Cognitive," and I was trying to explain how the cells always were sensing their environment and adjusting their metabolism and their actions according to what they were sensing. For example, a bacterium could swim up a gradient of a nutrient because it could sense the nutrient and adjust its movement accordingly. How far those kinds of cognitive behaviors extend is another thing that we need to examine experimentally. Some people are already making very sweeping statements about that, and they may be right, but we need to have empirical evidence to be fully scientific in our discussions of that. It's quite a daunting prospect to think about coordinating all that goes on inside a cell, all the chemical reactions, all the physical processes, all the growth and development, and keeping it straight. We don't find, for example, in the growth of a bacterial culture, we don't find cells dying off without an external stress applied to them. They're all getting it right. And that means that there's some kind of process happening which keeps things going in the right way. And we need to define those processes and understand how they work. And when we do, I think we'll have a whole different view of biology.
Absolutely. And obviously, that different view of biology will have repercussions far outside the life sciences, as you point out in your book. That'll have an impact in terms of economics, in terms of sociology, political theory, and just human nature, our understanding of human nature.
It will affect our approach to things like the environment, because we'll begin to distinguish between things which are destructive actions on our part and things which are there to maintain the stability and effectiveness of the biosphere. I think that's an important aspect of what this change will be. So that we'll have different outcomes to our collective activities as a civilization. And that can't happen too soon because there are too many people dying of heat shock.
Absolutely. Absolutely. So to end upon, the last question I have for you is your conclusion in this paper is that the many studies that have documented these capabilities in plants, animals, and eukaryotic microbes greatly expand our understanding of how living organisms can generate genomic novelties when evolutionary change becomes necessary. So, Professor, what future studies would you suggest researchers to explore in this area?
Well, I think what they can do is we now know conditions, how to create conditions in the laboratory which stimulate genetic change. So we can start to apply those conditions, and DNA sequencing is now so efficient that we can look at how they modify their genomes when they're subjected to these kinds of stimuli, which lead to genetic change. And we can also ask, if we change the conditions, the external conditions, do we change the genomic products that result? And that's a very, I think it will be a very fruitful way to approach this and to get some of this empirical evidence that these events are not random events, but they show some regularities. It implies you have to do the experiment that most people have now, and but that mindset is going to become more prevalent as time goes on, and I think we'll eventually see those experiments performed, and we'll then know whether or not we have hard empirical evidence to show that, as McClintock said, the genome is a sensitive organ of the cell.
Absolutely. Absolutely. That's a wonderful note to end this stream on. Professor James, it was lovely speaking to you again, and thank you so much for giving us your time.
Thank you.
Bye bye.
Sabur,
Yes.
has the first podcast been posted?
Yes, it has. And I'll send you a copy of that.
Okay, thank you. Or just a link would be fine.
Brilliant. Thank you so much,
Okay,
Professor.
thank you.
Cheers. Bye bye.
Cheers. Bye. All right, that's my podcast out of the way. Good.